Yeah, hello? That's it, okay. Okay. Hello, everybody. Welcome to the afternoon sessions. This session is about integrity and misconduct. The same rules apply as this morning. So we're going to start with Daniele Fanelli, who's going to talk about his research, summary effect sizes in meta-analysis after removal of retracted studies from the pool of primary studies.
Hello, is this, yes? Okay. Well, thank you for having me here. And the title pretty much says it all. This is a study about retractions and their effects on meta-analysis. Now, I'm gonna guess that the audience at a conference like this needs no introduction about what retractions actually are. And I bet many of you will be also familiar with the, by now, rather substantive literature on the prevalence and the nature of retractions, on their effects on citations. Now, what I found, however, quite remarkable is that even though I think most people instinctively think that retractions or assume that retractions are a problem, there are, to my knowledge, very few studies that have actually tried to look at what kind of problem retractions actually are for the scientific community at large. A notable exception that I thought was worth mentioning is this study from a few years ago, which tried to assess the financial costs entailed by retracting papers. And in particular, they looked at NIH-funded studies over a period of 20 years. I think I'm thundering a little. And as you can see, their conclusion were that overall, the total costs were about 58 million, which amount to less than 1% of NIH's budget, and the average cost per retracted article was rather substantive, almost $400,000, but you can see with a huge standard deviation. And in other words, I mean, depending on how you want to look at this figure, you could say that it's a lot because it's, in a sense, avoidable costs, or not as much. But certainly, one of their conclusions is that this is not a disproportionate cost compared to how relatively infrequent retractions are. And I think even more interestingly, as again suggested by the figures themselves, it's a cost that varies very much on a case-by-case basis. And in a sense, what we want to look at here is the same problem, but from an epistemic, a scientific point of view. What costs the retractions entail in terms of the beliefs that we have about specific effects or knowledge that we assume we have. And so very quickly what we proceeded to do was the following. We identified in the Web of Science all articles recorded as retracted to date, the date being last year, and identified all records in the Web of Science that were citing those articles, so that from these we could boil the number down to a list of plausible, potentially usable meta-analysis. And then we limited this list to last year, to 2016, and so we had 109 potentially relevant articles, and these were retrieved for inspection and included or excluded based on these few, and I would say rather, as loose as possible criteria. The main criteria being that we needed meta-analysis, which amongst the pool of primary studies had a retracted study within them. And this selection, and particularly this factor alone, reduced the list quite a bit. We ended up with 15 meta-analysis. In the abstract, we actually say 17, but upon secondary inspection, 2 actually couldn't be included either. So we have 15 meta-analysis, whose values were all converted to odds ratio and were coined, meaning that we reversed the values whenever necessary to ensure that the direction of the treatment effect was always the same. And then we did something very simple. We calculated the summary effect size for the meta-analysis as given, and then recalculated the value without the retracted study, and took a ratio of the two. So when the ratio value is 1, there is no difference. When the value of the ratio is above 1, that means that the retracted study was presumably falsely increasing our confidence or our belief in a certain effect, and vice versa, when the ratio is smaller than 1, it was actually decreasing it. And, well, with no further ado, these are the overall results. So what you see here is the ratio of odds ratio for all these studies. And these are listed, as you see, on the left column, not by the author of the meta-analysis or the title of the meta-analysis, but by the author of the retracted study that was included in them. The forest plot in the middle is not going to say much this way. I regret actually not having log transformed the scale, but you get the numbers on the right for this ratio of odds ratio. Now, two things to notice. First of all, that in 2 cases, again, the abstract says 3, but 1 was removed. We have the same paper being cited by 2 different meta-analyses, which already says something about the potential impact of a retraction, which is not just about 1, but potentially several studies. And the other, concerning the values, as you can see, is that overall, the values that we get are, first of all, for the majority above the value of 1, meaning that generally speaking, on average, removing the retracted study led to a decrease of the estimated effect sizes. But also you might notice how in most cases, the values are actually very close to 1 anyway. They're very, very small, with a few exceptions. And so the overall pooled summary that you see at the bottom, so this is a meta-analysis of these values, assuming random effects, which sort of tries to give more of an equal weight to the different studies, but is very, very small. It's 1.02, so at best that would be a 2% higher value for a meta-analysis with a retracted study, and in any case, the confidence interval is, of course, very, very large. Now, how can we explain, in case for the people at the bottom, this is the overall figure of the pooled results. And so there are 2 observations that we can make about what can explain some of this variance in results. One, which is sort of predictable, is that the impact of removing a retracted study is higher for smaller meta-analysis. And so here you see, of course, the values of the ratios of odds ratio plotted against the size of the meta-analysis, the number of studies included. The other observation, and this is sort of an exploratory observation, but as I'll try to illustrate, I think it's quite interesting, is that there seem to be differences concerning what the retraction occurred for. Now, the caveat here is that this classification was based on the text of retraction notices, and as we probably all know quite well, these are not always reliable information, particularly when it comes to scientific misconduct, because there are understandable concerns on behalf of editors of making the case too explicit unless there is no risk of a legal consequences. Nonetheless, based on what the retraction notices declared, we have this whole group of retractions due to plagiarism or to other, I'm saying QRP, but really this is other ethical breaches of research practice that have nothing to do apparently with the data or the results. And with one possible exception, the values are all very, very close to 1. There are 2 studies that were retracted explicitly due to fabrication and falsification. Now, you might notice that the first author is the same, the case of misconduct was the same, but the articles are different. And in both these cases, actually, the values are quite extreme and above 1, meaning that essentially these articles were presumably overestimating and leading, therefore, the whole literature to overestimate the value of the result. But equally interestingly, I would argue, the retractions that were due to honest errors, seemingly, so errors that had to do with the methods or the data, but were sort of openly declared by the authors as being a mistake, are, at least in 2 cases, extreme in the opposite direction, meaning that these studies were actually bringing the overall summary originally down, and without them, the estimate goes up. And so, just to quickly summarize the study, again, we collected this, for now, small pool of meta-analysis and looked at whether removing the retracted paper made a difference. And the results are that, on average, across these studies, we cannot pick really a statistically significant direction of effects, because there is, again, quite a remarkable variability in what specifically happens. And on a preliminary or exploratory level, we can conclude something nonetheless about the fundamental question: what does a retraction make in terms of a difference? And there's probably, on average, very little, but that's because, quite similarly to the estimation of the financial costs, this is going to vary on a case-by-case basis. First of all, because smaller literatures are going to be more impacted by a retraction, but also because in the case of fabrication and falsification, we seem to observe, and it seems plausible to hypothesize, that the overall summary estimate might actually go down. So we might be misled in believing effects or treatments when in reality this is based on false and made-up data. But also, when there is a genuine error that is being rooted out, then the exact opposite is actually the case. And so, a retraction in that case, you could argue, improves our knowledge and our confidence in results. And, well, this is all I had. I'll thank Julie Wong, who helped us collect most of the data, and I'll thank you all for the attention. Thank you.
Thank you. Are there any questions? Can I just ask a point of clarification? So, in this study, were the retractions already in place? What was the timing of the retractions compared to when these studies were included in the systematic reviews?
That's actually a good question. We didn't explicitly look at when the formal retraction was issued. There's no question that the retractions were recognized, and all these meta-analyses had been published last year. Presumably, that would entail that the retractions, that's actually what I was assuming, but you just made me think that maybe in some cases the authors had actually missed the fact that the study was retracted, and we'll look into that. Thanks.
Okay. Question there?
Ivan Oransky from Retraction Watch. That was my question. Thank you, Jagisha. It was one of my 2 questions. I guess the other was, I just want to make sure I got this, because I may have just been unclear, but the 15 was in the last year, or?
Yeah.
Okay, so these were just in the last year. So you weren't including anything like Joachim Bolt from several years ago, because.
So evidently not. That could have happened, again, presumably if the meta-analyst had missed the fact that these studies had been retracted. But the meta-analyses were published last year.
Okay, yeah, because that was a case where Njama republished the meta-analysis without, did exactly what you did here and sort of took it out, but that was a few years ago, so I just wanted to make sure I was clear. Thank you, very interesting.
Question there?
Andreas Lund, Center of Evidence-Based Medicine in Denmark. It's a little bit similar to the question you asked before, because I think what you were asking was, here in 2016, what does it matter for those meta-analyses created now, where we know that the flawed papers were actually retracted? But I think an interesting question would be to look at the time window from when it was published until when it was retracted, and to see actually what did the data contribute to in a meta-analysis performed at that time, where we didn't know that the study was flawed. Because I think today, if you know a study was flawed, you would probably not include it in a meta-analysis, or at least if you do, you would judge it as high risk of bias. Whereas in retrospect, you might think that it could actually be a sound study, and therefore it could contribute to an invalid conclusion. I don't know if you get my question.
So if I get your question, you're saying a study that is flawed, but we don't know yet, might have an influence on the literature.
Yeah, because it could be that it was a big trial, and then you would conclude that no new trials are necessary. But since you know it's retracted, additional trials might have been done in order to kind of answer the questions.
So that would be another contribution of a retracted study or a retraction to the overall research process. Is that what you're...
Yeah, I think just the interesting question is, at the time the study was published, did it actually affect the literature? And what you are asking is, here in retrospect now, when we know it's retracted, does it really contribute to anything? And I think it's maybe not the clinic, from a clinical point of view, I would like to know whether I was fooled at that time.
Right, I mean, so yeah, I think it's an interesting point in a sense that that is also a possible contribution that even flawed or even fabricated studies might make. And again, another sort of controversial argument, if you like, comes to mind concerning scientific fraud even, the fact that sometimes the very fact that someone dared, purely because they believed something to be the case, to make up some data just to make the claim first, stimulated research in a way that paradoxically becomes positive. And I think the observation you're making does tap into a little bit of what the initial, again, preliminary moral that can be drawn from a study like this is, which is that retractions can have varying effects. I think, and this is generally my position, and again, it's sort of somehow reinforced by these results. You know, I could be biased in this way, but this is increasingly how I see it, that we sort of stigmatize retractions as something that is essentially just a problem to be removed, and that in an ideal world, retractions would never happen. Whereas in fact, the picture is more complex, and we show there are at least as many reasons to celebrate at least certain kinds of retractions as there are to stigmatize them.
Drummond Rennie, San Francisco. I know that this is not your message. Your message is that if you do meta-analyses, this is what happens. And I think I like this paper, and I hope you'll continue and continue and continue until you're as old as I am. It'll be interesting to see. I don't want anyone, perhaps no one is, getting the impression that this doesn't matter, which is a different thing. And it's not what you're saying. And that has been used as an excuse for not publicizing retractions at all. I think of one of, say, 70 or 80 that I've seen and had personal experience of, and before Ivan was born. For example, the big breast cancer study, where a Canadian center of the study effectively made up all the evidence. It was taken out of the study, but the journals were not told at all. Articles or manuscripts, including those centers, with known to be defective data, were still submitted to journals during that time. And it was only when the newspapers got hold of it that everything unraveled. At the same time, there are huge consequences on the researcher and everybody down the line, their colleagues. Institutions can be brought to a halt and nearly bankrupted by this sort of thing. It matters. That's not what you're saying, and congratulations on a nice start.
Great, thank you. And let me pick up on that a second, because I think this goes right at the core of the issue here, which I think that we should start always keeping in mind clear distinctions between types of retractions. There are retractions that are due to reasons that are really bad and that are demoralizing for the scientific enterprise, and that in general will cause the kind of problems that you just illustrated. There is increasingly, in fact, a second form of retraction, which has to do with authors doing the right thing. And there is, I think, a growing understanding that this kind of behavior needs to be encouraged, and that although it might cause some problems for editors, surely, it is something that is vital to the research process. And it's something that, it might seem paradoxical, but needs to be rewarded and encouraged. And so that's really is the core. I wouldn't disagree with you, but again, I think you're thinking of the bad ones, which have a bad consequence also for meta-analysis and the literature. But there is a whole universe of possible other flaws that somewhat are inevitable, and that the correction of which should be honored.
Okay, if there are no more questions, I take it there are no more questions from Twitter or Facebook? Can't see. We'll move on, so thank you.
Thank you.
And next we have Christina Bennet, who's going to talk about assessing the outcome of introducing a digital image quality control review into the publication process for research articles in physiology journals.
All right, I'd like to first thank the organizers for this opportunity to share with you our analysis of our Society for initiating a quality control check for images prior to publication. Our study sought to assess whether the QC check was effective in identifying and correcting images with modifications prior to publication, and whether returning authors who received a QC check once were repeat offenders. The American Physiological Society publishes 15 journals. What I'm going to speak on today will focus on the 7 AJP titles. These are primary research journals, and they combined publish about 15 to 2,000 articles each year, or 1,500 to 2,000 articles each year. And many of our authors utilize photographs to represent their data. These photographs often are images of cells, tissues, organs. They often show expression of DNA, RNA, and protein in the forms of blots and gels. And many, I would say several years ago, our art department, who we have in-house, started to notice that there were image modifications within the figures that they couldn't tell whether they were appropriate or inappropriate. Around that time, the Journal of Cell Biology, at that time led by Mike Rossner, provided some guidance for journals to look for images and to see what types of modifications were within them. We updated our information for authors and gave guidance to our authors that we would look for evidence of splicing, or that is rearrangement of a particular photograph, extreme contrast adjustment, which is either altering the brightness or contrast to an extreme. We'd look for selective editing, which is altering a portion of an image that isn't done equally across the entire photograph. And we'd look for duplication, either using the same image to represent different experimental results within one figure or somewhere within the rest of the manuscript. As I mentioned, we do have an in-house art staff, and so they utilize the forensics tools that were developed by the Office of Research Integrity at the Department of Health and Human Services to get a forensic, kind of like a fingerprint of photographs to see how authors prepare their data. Because this was an art staff role, they began to do these checks as they were preparing the figures for final publication. However, when concerns were identified and corrections could be made, it would result in us having to post some type of core agenda because by the time the figures are being prepared and analyzed, the article is already posted online as an early view publication. This was frustrating to both the authors, who were making somewhat possibly minor corrections, and to staff, because they were doing an extra step within the publications process. So we moved the QC check to after acceptance. And so now our authors receive the acceptance letter, and the manuscript file moves into a new little box within the manuscript submission system, which allows our art staff to do the assessment for image modifications prior to the article being posted online or final published. And this allows us to get revisions to non-fraudulent modifications prior to publication. So the data I'm going to talk about today spans over from 2009 to 2016, because it took us several years to initiate this QC workflow. And as you can see, in 2009, there was no formal workflow, and this was about the time when the art staff was noticing modifications, and our leaders at the APS, including our director of publications and the publications committee chair, realized that we needed to take a closer look at the images prior to publication, and we needed to find a way to do this quickly so that we would not delay the publication process. And one of the things that they added was my role as an ethics manager to handle these types of queries. I do have a background in research, and I could look at the science, whereas our art staff could look at the modifications. And if we thought we needed to seek corrections, there would be somebody in-house to answer the scientific issues related to the concerns. We also saw this as an education tool. Our template letters that we use go out to the corresponding author. We notify them of the concerns that we've identified. We state what our information for authors guidelines note about that possible error, and then we ask for corrections along with the original capture. If they provide a correction, and if they provide the original capture and it shows that nothing has scientifically changed and it was just a minor modification, then we allow the correction to go through. So it took us several years to onboard all 7 journals, but by 2013, all of the journals were within the workflow. We had a banner year in 2011, so out of approximately 2,000 articles that were published that year, 8% of them received a QC letter from us asking for corrections. However, since then, year on year, we've had a reduction in the number of QC cases for image corrections. One of the questions we wanted to look at was how we had fared with the request for core agenda versus giving the authors the opportunity to revise prior to publication. And as you can imagine, now that the workflow was, once it became all in place, our number of core agenda went down significantly. So much so that in 2015 and 2016, we did not miss any articles up until so that they did not need a core agenda. All of them were, if revisions were appropriate, they were invited to revise their work prior to publication. And that's not to say that we allow revisions for all concerns that are identified by this QC check. Instead, we do identify concerns every year that are serious enough that we rescind the acceptance and issue an ethical rejection. These include findings that the images were either falsified or fabricated. And in those cases, those are the red bars. We've retracted articles at the AIPS version for fabrication and falsification. And we've also rescinded acceptance and issued ethical rejections for articles where they could not find the original captures, and we weren't convinced that something untoward didn't happen, or the data has changed in some way such that it requires them to repeat the experiment and go through review again. Another question we wanted to look at was whether our types of image issues that we identified had changed over time. So as I mentioned, we looked for evidence of splicing, extreme contrast adjustment, duplication, selective editing, and compound meaning a combination of one or more of these issues. In 2009, our art staff identified many issues of splicing, and that was the major thing that they were looking for. However, as time went on, they began to get more savvy with identifying that extreme contrast adjustment was another way to mask modifications. And so now it's quite common for us to identify errors related to splicing and also extreme contrast adjustment. And we always have our handful of duplication and selective editing, but those vary year by year. And then another question, because we made this, we wanted this to be an education tool for our authors and wanted our template letters that went out to authors to be a way to educate them on good presentation practices. We wanted to see if our authors who received these queries and submitted to us again were repeat offenders. So we took a snapshot from 2013 to 2015. This is when all of our journals were now in the QC check process, and looked at whether those authors had submitted again. So out of those 326 authors who received one QC query, 58% submitted another article by the end of 2016. And only 8 out of 190 of those were involved in a subsequent QC query. And we like to think, although this can't be fully verified, that our authors are learning. I think by being part of a scientific society and having a member and good author base, we do have an opportunity to educate our authors and to encourage them to do better practices. And the other caveat I will say is that I did not look to see whether their second paper contained the same types of images as were in the original paper. So in conclusion, implementing this QC check to review image modifications in accepted articles has resulted in the reduction in the number of post-publication corrections. So it makes me sad that we don't often hit retraction watch as much as we could. And the publication of more images as they were originally captured. We do detect serious errors and fabricated data now prior to publication. And we do support our authors by educating them on the appropriate digital image presentation practices. And what's encouraging is that we are seeing a decrease in the number of QC queries year on year, which suggests that our returning authors do adhere to these guidelines, and we look forward to tracking this data further. Thank you.
Just see who gets to the mic first. Okay, go ahead, Ivan.
Thanks, Ivan Oransky, who was apparently born after 1994. But, I'm delighted. My internist will be even more delighted. Really interesting presentation. Of course, you wouldn't be surprised to hear me say that. And I don't know that there's a way to actually look for this. But I'll ask, and you can tell me there wasn't. Do you know what happened to the author? You said 58%. Obviously, a lot of authors came back even after the QC check and what have you. Do you know what happened? You know, overall, did everybody who had a QC check come back? I'm not articulating the question well, but did fewer of them come back than you would have expected, based on sort of typical patterns of submission to your journals?
Yeah, so I didn't look at the trend of non-QC check authors versus QC check. But within a 4-year span, having 60% submit a second paper seems pretty reasonable for biomedical research.
Thank you.
Chris Papazian, Kansas City. So for those manuscripts that you ultimately decided that there was some serious problem with the image due to fabrication or inappropriate manipulation, aside from just rejecting the manuscript, is there any other action that you took with those authors? Yeah,
so we do present their cases to our publications committee to determine whether authors shouldn't be uninvited from submitting work to our journals for a period of time. We also informed the institution many times about these issues.
Becca White, Journal Neurosurgery. We don't deal with these issues at the journal where I am now, but before I was at a stem cells journal, and we saw this quite a bit. And we're a very small journal with very small staff, and so our department was one person. And we certainly couldn't afford to have a full-time staff member who was in your role. So what would your suggestion be to smaller journals? Because this is a huge issue, and you do need someone who is trained in the science and in the art to be able to be doing these checks.
Yeah, so, one of our concerns was putting burden on the peer reviewers, who are also scientists, to do any of these types of checks. But there is something to be said about having a scientist know what the modification is and whether it's worth pursuing or not. So one of the things that we initially told our editors and editorial boards was to use the PDF and zoom in. Maximize the photograph to 400%. So when you're looking at the figures, if things are duplicated, if things have hard lines through them, they should be visible to the naked eye. The forensics tools are fantastic, but we don't want them to become investigators during peer review. They need to focus on the science, but ask them to zoom in. I mean, it actually sometimes frustrates me that something went through peer review and got accepted and no one saw some of the modifications.
I'm Barbara Goldman from American Society for Microbiology Journals. Christina, thank you. That was a very interesting presentation, very valuable. At ASM, we do very much the same thing and have had a similar experience. The challenge that we have now is that the ORI forensic tools are terrific for detecting pixel differences from figure to figure, image to image. And the challenge has been in blots and gels, duplication of bands, because they all look the same pretty much. But there are some amazing individuals with visual acuity that is astonishing, who are able to scan figures and recognize duplicated blots within hundreds of different experiments, including from one paper to another. And I'm just wondering if you have had any experience in tools that help you identify the duplications, which we really have not been able to find. Yeah,
so we do rely on the forensics tools. Our art, so I do some screening at times, but Eric Fanelli, who's our editorial art manager, has Eric eyes, as I call them, and he sees duplications like nobody can. So it is something about being trained in looking at images. The more and more you look, the more and more you think, oh my gosh, that looks really similar, and it looks even more similar, and you just get the eye. So as practice, the more practice you do, the more better you'll be able to see the slight changes. And, it's biology, so every blot should not look the same. And that's when you do, it's when you use the forensics tools just to make a slight modification, you start to see the similarities come out. But it's not easy. Yeah.
Well, hello, Jennifer Byrne from Sydney in Australia. You're probably familiar with the study that was done by Elizabeth Bick that I think was published in 2016 in MBio, which looked at a very large number of publications, and I think identified differences in geographic distribution of publications that included problematic images. I was wondering whether you saw any evidence of that in your study.
We don't usually assess it by image modification, but overall we tend to have a 50-50 US versus non-US, breakdown of ethics cases. So I don't think it's one country or, one area that's the biggest offender for these types of issues.
Hi, Juno Shiro, Mayo Clinic. I was wondering, you mentioned duplication, and I think from what you're saying within a single paper, but is there an easier way to look at, for example, comparing, what's in the current paper versus the author's body of work that came before? Is there some non-manual way to do that?
If you can rely on PubPeer, which seems to be much better at, having sleuths look at a corpus of work. But no, I think our peer reviewers identify duplication better when it's throughout a body of work than we do, because we aren't going back to doing a whole, and we focus on the article that we have. Right. And I wish we could. I wish we had a database that we could put all of our images in and find them again if they're being reused.
And is your in-house process, is that automated, or is it still people looking at individual images running individual tools?
No, it's still manual. I mean, we use tools, but that's why it took us several years to get this QC check incorporated,
Sure.
because we did not want to slow down our publication process. And it takes training of, whoever's doing that to get good at it and quick enough to go through these images within 10 minutes versus an hour, which is when we started. It took forever.
Okay, thank you.
Yeah.
Hello, Michael Todd, Innsbruck, Austria. Forgive me if you answered this during your talk, but I'm coming at this from the medical writer perspective. And I do edit images when with papers, never unethically, of course, but I'd rather it be clear and not be asked to address the ethical issue. So if I'm submitting a paper, should I always tell the journal? What would you like to see from the medical writers? Should we tell you we edited an image?
Yeah, if authors do a non-standard modification, it would be good to use the legends to say that I'm presenting 6 out of the 12 lanes for this particular image because these are the ones that are pertinent for the study, and I'm indicating this with a space. Or I've extremely, my photograph has extreme contrast to highlight this one aspect. I mean, tell us what you're doing. Otherwise, it may look like you're trying to sway the data in a way that you don't really intend to do so.
So I just put it in the legend that it was
Yeah,
edited. All right.
that's what we encourage, yeah.
Okay, if there are no more questions from the floor or online, I do have one more question for you, sorry. Just related to that last question, you mentioned that over the years the authors have become better at knowing what they can and can't do. Do your instructions for authors provide detailed guidance for what can be done and what isn't acceptable?
I will say that they're more of can'ts, that they can't splice without declaring it, they can't over-adjust, but we do encourage them to declare more in the legends if they do need to do some form of modification.
Okay. Thank you. Okay, so next is Cyril Labbe, and he's going to present fact-checking nucleotide sequences in life science publications, the Seq and Blastn tool.
Hi everyone. So this work has been done with Jennifer, who is here in the audience, and she's a biologist from Australia, and she will join me for the question, because I'm sure there will be plenty of things that she will be able to answer, and I will not, because I'm a computer scientist from France. So you will have to struggle with my French English. I'm sorry. I will try to speak slowly so that you can catch up the deep meaning of what I try to say. Okay, so this is about automatic detection of questionable research paper. So I think you all know what we are able to do for plagiarism using what is behind the detection is hashing function. But there is also work that are dealing with detection of automatically generated paper. So this is work that is funded by Springer Nature in my group. So we are using the intertextual distance to do some machine learning to detect automatically generated paper. But these kind of tools do not really detect the fact that data are manipulated or fabricated or whatever. So we would like to have something that help to find out errors. So as you have heard before, there is no tools to find image duplication or things like that. So we won't try to do that. We'll just try to extract facts from the text and try to see if what is said in the text is true or not. So it started with 5 publications that Jennifer saw. And so Jennifer not discovered, but well, let's say discovered the particular gene. And all those paper was speaking about this particular gene, and there was what she called obvious error in this paper. So all these 5 papers were really looking alike. There was same kind of figure, no duplication of figure, but same kind of figure, same section, more or less the same wording. Wording were different, it was not plagiarism. There was some kind of plagiarism sometimes for some paragraph, but really rephrasing. So that's why I guess they were not caught by a cross-check or what has been used as plagiarism detection. And they were all ending with kind of strong affirmation about the use of this particular gene regarding different kind of cancer. So I asked Jennifer what kind of use errors there was in. And so this is the thing that she was able to explain to me, and I will try to explain to you, but if you have questions, you will have to ask it to her. So, for example, here there is sentences and gene and DNA sequence that are used as targeting sequence, the green one, and you can check that this particular sequence is really targeting this particular gene using a Blastn, which is a software that is done by NCBI. Well, it's a very famous software that can be used to check that a particular sequence has homology with the human genome. So it goes, what does this software? It goes through the human genome and check the homology of a particular sequence. And so you are able to check that this green sequence targets a human gene that is said to be targeted in the text. But for the red one is said in the text that it does not target, it does not have any homology with the mammalian genome. But if you do the Blastn search, you will find out that there is a really strong homology with the human gene. So here, there is something, a fact that is said in the text that is completely wrong. And this is this kind of check that we will try to automate as much as possible to help people like Jennifer to find this kind of problem. So how does this tool, Thick and Blast, works? First, you look for all the sequences in the text, and you analyze each sentence to find out if the sequence is a targeting sequence or a non-targeting sequence. And you end up with a status for each sequence. Then you perform a Blastn analysis automatically for each sequence and get the result from the Blastn. And then you do a comparison between the fact that has been extracted from the text and the fact that come from Blastn. And then you are able to tick if fact to know if it's a green one or a red one, if there is a problem, or if what is said in the text is in coherence with what we know from Blastn. So we have used 2 different corpora to assess this method. So the first one is composed of 48 papers that we know that they are problematic. We know that there is plenty of redundancy between them. They are all dealing with a knockdown problem, knockdown studies. And we know that there is a certain amount of them that are not using properly DNA sequence. And there is a corpus of onion paper in which there is plenty of papers that has been retrieved using the PubMed similar function. So you just click on a paper and you get papers that are similar to the paper you clicked. This corpus is mostly composed of open access paper because you cannot scroll fee-based database. So whatever are the results, you cannot interpret them between open access and fee-based access. So I will just jump over this slide. it's about, yeah, there's been some change here. Okay. So what does Sync and Blast is an automatic extraction from the nucleotide sequence using techniques that are named entity recognition. Okay, so that's computer science techniques based on rules and thesaurus. Then you perform the Blastn check, and also the status of the sequence is extracted using a finite state machine and, well, some different kind of thing, and then the Blastn analysis. And the results are, so we were able to extract sequence for the 48 paper in the corpus, in the problematic corpus, and only 73% of the paper in the corpus, in the unknown corpus, were containing DNA sequence in them. So sometimes Seek and Blast is doing mistake. So for example, in the corpus P, where we know there was problem, we didn't identify correctly the target, if the sequence was targeting or non-targeting, for 19 sequence out of 1,300 and more. Okay, so you have to know that this tool is doing mistake, and so there is a manual screen that is needed after, just to check that what Blastn is, Seek and Blastn is telling is true. So, at a conclusion, so we did a kind of automatic detection, fact-checking, which, as you know, is quite à la mode today. Something that is very useful for fact news detection, for example. But often fact news detection are based on the source of the news. So, for example, here, you will say that a paper might be fake because it comes from China or because it has been published by this publisher or things like that. So it's obviously things, or because it comes from this university or whatever. So maybe this is not a good way to go. And there is also a trend to try to detect statistically flawed papers. So there exist some tools that extract numbers from the papers and try to see if the p-value is good and things like that. So we are going a little on another track here. Let's try to find out the facts in the text and try to check the fact. And so there is an online prototype that is available at this URL. And the next steps will be to try to avoid as much as possible the false positives that we have in our results. And so this could be done by some techniques like more in-depth analysis of sentences. And how this tool could be used, well, there is many ways to use that, but this must not be seen as a way to fight misconduct, because, well, this is quite just kind of patch. You will not find the root of the misconduct. It's only what you see, the results of the misconduct. The root, we think, are more related to the pressure for publication and things like that. Okay, thank you very much, and I will ask Jennifer to come to help me for the question.
Thank you. Are there any questions from the floor?
Do people understand it?
Well, we do. We have one questioner. So, go ahead.
Chris Papes in Kansas City. I'm curious as to how specific this tool is as far as looking at a nucleotide sequence, because I co-author lots of papers that have nucleotide sequences on it, but I'm not involved in doing that particular aspect of it. And I suspect if we have 18 authors on a paper, probably only 1 or 2 actually know the sequence, and nobody, and it's, you know, it's mind-boggling to put that stuff in place and not screw it up. So how do you distinguish between somebody who just blew it versus something that is actually fabrication?
Yeah, I think that's a great question. So, I mean, I guess I came to this field because I write these kinds of papers, and I'm very aware of how difficult it is to control the facts when you are dealing with nucleotide sequences. So in terms of the analysis, I think Cyril presented about the 2 corpora. So one is this corpus that we have published in the journal Scientometrics, and these papers are very uniform in terms of their structure, what they're purporting to do, and the kinds of errors that we identify within them. But in the corpus U, which is in the larger corpus that we're still analysing, what I have found in that corpus is that there are different kinds of errors. So as an example, I can think of one paper that might have included RT-PCR sequences for about, say, 10 genes, and one of those sets of primers targeted a gene other than the authors described. So the way that I interpret that as a working scientist is I think that's possibly just sloppiness, particularly as the gene that was targeted by the sequences is related to the one that was written in the paper. It's not the same gene, but it's of the same family. The kinds of errors that we are more concerned about are the really very blatant errors where, for example, a non-targeting control, a sequence that's stated to be a non-targeting control that should not target any gene, very simply, it's very simple to demonstrate that that does in fact target a gene. So that kind of error, I think, is more fundamental and more difficult to be explained by sheer sloppiness. But I agree that, I mean, I guess as a scientist, whenever we read papers, and if there are sequences that are particularly relevant to the conclusions of those papers, we will blast them very routinely. So I do this kind of thing all the time. But I'm not sure that everybody in the community will be using those same practices.
Just would add the fact that even if it's just sloppiness, there might be a correction to publish because there was an error somewhere. So even if a retraction is not needed, just a correction will be needed.
Are there any more questions?
Hi, Constance again. I hope it's okay for me to ask another question. So I did cancer research. I was a lab manager of a cancer research lab for a few years. So I can imagine all the possible ways that things can get screwed up. So I'm just wondering, like before we publish a paper, we have people to check out the comma or like the format, but there's no one check basic genetic sequence, whether it was correct or not. Is kind of interesting, right? What is more important, a comma or whether you're using the right gene? But I'm actually just really curious because I was reading the conflict of interest disclosure that Cyril, are you actually, it sounded like you're trying to find a method to detect automatically generated scientific paper. So are you trying to optimizing the detection of automatically generated paper?
Yeah, that's it. Because there has been some papers that were automatically generated that were published in high-profile publisher, and so that a tool was needed to detect them automatically because they were published and sold. So, and the tool is actually running, and publishers are using it. It's an open source tool that is available.
Hi, I'm Melissa Vaught from contractor at NCBI in Maryland. So as I understand, your tool is designed so that you have to have an open access HTML publication for you to be able to do the data extraction. And my question here is, what about the issues of a lot of data now, especially even the methods being moved into supplemental materials, which are often in PDF formats, which really create a problem for the data extraction and how you can approach that?
Okay, so we've got we've got some false positives that come from the fact that we are working with PDF. because we extract the text from the PDF, and so we lose all the formatting thing. So for example, all genes that are in table or things like that, we are not able to extract them properly. And some of the false positives that we have come from this kind of problem. So if we were working on HTML file, it will be really more easy. And so I think that if we work with the data that publishers have, for example, it will be more easy and we will have less false positive.
I might just add that, a related issue is that there's no requirement to actually publish nucleotide sequences within publications. So the reason why we were able to find some of these papers was because the sequences are there, but particularly negative control reagents are often not defined. So I think our work indicates that probably there needs to be some guidelines in terms of requirements for people working with these kinds of reagents that can be verified using the sorts of tools that we're developing.
Thank you. Tahir Pillay, Journal of Clinical Pathology. I'm a molecular biologist and I've worked with cloning and expression projects. One of the things that I've learned about in going to many conferences was that sometimes I've heard that some of the mistakes could be deliberate because in a highly competitive field, you really don't want your competitors to be able to reproduce your experiments quickly. I'm not sure if that's really part of the problem.
You know, that's something that I hadn't considered, but I think it's a bit analogous to my grandmother, who apparently every time someone asked her for a recipe, she would never give you the full thing, so I think it's possible, but I think the stuff that we're studying is probably not that deliberate, but I agree that could be an issue.
Okay. Thank you. We need to move on now, so thank you, Cyril. And now we have Stephanie Boughton, who's going to present types of research integrity issues encountered by a specialist research integrity group.
Hello, so I'm going to be presenting some data on the types of research integrity issues that we saw as the Biomed Central Research Integrity Group. I'm now actually part of a wider Springer Nature Research Integrity Group, but this data is all from BMC journals. So research integrity issues is quite a broad, sort of a broad category of things, including research ethics issues with publication ethics, but there's not a huge amount of data on how common they are. So in terms of published literature, there's some data from retractions, so the reasons for why articles are retracted. There's also data if the results of institutional investigations are made public. There's also surveys asking sort of researchers how common misconduct or questionable research practices are. And then in terms of sort of in getting an idea of more day-to-day things, we see from the cases brought to COPE forums, we get an idea of some of the issues that editors are dealing with, but obviously these are only the really complex cases that the editors felt that they need to go and seek advice from COPE. But we know little about the day-to-day research integrity issues that are faced at journals. So we decided to have a look at the data on the research integrity issues encountered by the Research Integrity Group and to categorise the relative frequency of the different types of issues. So the Research Integrity Group is a specialist editorial group. We provide support for editors and in-house journal staff in dealing with issues relating to research ethics, publication ethics, and research integrity. And at the time of data collection for this study, we provided advice across approximately 300 open access journals spanning biological and medical disciplines. And so the way that we hear about these issues and provide advice is we receive emails from internal staff or sometimes external academic editors asking for advice on research integrity issues relating to submitted manuscripts or published articles, and these really vary widely in terms of their complexity. And the amount of support that we provide then varies accordingly, so from providing advice to the editor to taking over handling and helping them with complex cases. So for this study, our inclusion criteria were all new queries that were sent to the BMC Research Integrity Group over a 2-year period from the start of January 2015 to the end of December 2016. And these related to any aspect of research integrity and in any journal published by BMC on submitted manuscripts and published articles. And then we categorized these issues that were arising by the type of issue they were. And then we've done some analysis on the categories, looking at the overall frequency of these different types of issues, and then looked at them pre-publication and post-publication. And these are sort of queries that sometimes may amount to nothing or may turn out to be, say, a serious issue with the manuscript or article. So we based our categories on the Cope case taxonomy, but these have been adapted to fit the types of issues that we see on our journals. So authorship is basically anything relating to authorship. So, for example, authorship disputes, maybe sort of questions of whether there's been sort of authorship for sale or gift authorship or ghost authorship. Competing interests is any like allegations of undeclared competing interests. Data issues we've used as a very broad category. So this is anything pertaining to the content or the ownership of the data of the paper. So, for example, a data ownership dispute, honest error found in articles, or maybe cases of alleged fabrication or falsification of data. And this category also includes concerns about image manipulation as well. Ethics and consent is any questions relating to ethics approval for the study, concerns about the ethics for research involving humans or animals, concerns about the consent process from the study, consent to publish potentially identifiable information or images, and we've also grouped into that any questions about trial registration. Peer review process, any concerns about the peer review process, and maybe any concerns about potentially unverifiable reviewers or attempted manipulation of the peer review process. And then plagiarism and duplicate publication also includes duplicate submission, text recycling, and use of images without appropriate permission. So during the time period that we studied, we received 1,040 individual queries from editors. And when we look at how these break down in terms of pre-publication and post-publication, we see that the largest group is relating to submitted manuscripts, so before publication, and there were 690 of these, so 66% of the total queries. And then 350 of them, so 34%, related to published articles. And as we can see from this chart, ethics and consent was the largest category, followed by plagiarism and duplicate publication. And just to put some numbers on those, ethics and consent amounted to 362 queries, plagiarism and duplicate publication, 240. But if we move to look at these pre and post publication, which I think is more interesting data, we see that before publication, almost half of the queries that we're receiving are relating to ethics and consent, and that the other categories are then much smaller. But if we then look at this after publication, so these are the queries that we as a team are receiving relating to published articles, we're seeing that ethics and consent has gone down to a much smaller percentage of the queries that we're receiving, and the category of data issues is the most common, amounting to 41%, which is 144 queries. And it's worth noting, just with these two charts, that the overall number of queries received on submitted articles was a larger number. So actually, in terms of absolute numbers, everything is arising more commonly on submitted manuscripts, so pre-publication, apart from data, where we're getting more queries on published articles. So From the results, we can see that some types of queries that we're getting are arising in different frequencies in terms of whether they are before or after publication. So ethics and consent is amounting for by far the biggest group before publication. And I think there's a number of reasons for this. I think this is coming up before sort of on submission, because authors are required to put in statements of ethics approval, of consent, in their manuscripts, and editors are checking these as they're assessing the manuscripts. And obviously reviewers and editors are also alert to any concerns about ethics involving humans or animals. And I also think that perhaps the reason that we're getting many of these escalated to our team is perhaps the sort of perceived seriousness of these issues, and the fact that maybe the editor feels that they need some advice because of the fact that this could be concerns about research involving humans. Now, some of these queries sort of vary in terms of concerns about something that's maybe seems like something very unethical has been done, to fairly simple queries about, I'm not sure whether this ethics statement is appropriate. When we then look post-publication, we saw that data is the biggest issue that's arising. And I think, again, there's a number of reasons for this. I think, firstly, if there's issues to do with, say, data ownership disputes, I think the person who's disputing the ownership of the data will notice this when the article is published. I think in terms of concerns about the content of the study, I think, again, the reason that these are coming up more frequently after publication is perhaps that that there's more eyes on the papers, that people are trying to reproduce the studies, and that's when concerns are being raised. It could also be that the editors, if concerns are raised about the data prior to publication, then they're rejecting the manuscripts because they feel very happy dealing with that themselves, whereas if concerns are raised after publication, then they're coming to us for more help. And we do see we provide support on dealing with retractions and sort of serious corrections, so this may be why we're seeing more of those. Other issues seem to come up similarly before or after publication, and say, for example, authorship was relatively low compared to maybe how much you'd expect there to be sort of issues with authorship, so maybe again it's that editors are comfortable dealing with these issues on their own. Now, there's quite a few limitations to bear in mind when interpreting this data. There's no requirement for editors to escalate issues to the Research Integrity Group. We're there to provide support as and when they need it. So it may be that the editors are dealing with many issues themselves, or actually their in-house contacts are maybe comfortable dealing with particular issues. Not all of the things raised to us turn out to be issues, and we're seeing a wide variation in complexity and severity of the types of issues. And so some of them may relate to just one manuscript, but then some of them will relate to multiple manuscripts, which could span different journals. But these will be categorized, the way we've analysed our data, these would be categorized as sort of one query. And all of these journals are biomedical journals, so we couldn't necessarily extrapolate this data to other fields. So finally, some thoughts to end on. I think there's potential implications from these results on informing the training and resource needs for editors and in-house staff, so the areas that maybe they feel less comfortable on, the areas where issues come up more frequently and that more training and resources is needed. But perhaps, for example, with the number of ethics sort of issues that are coming up pre-publication, perhaps more training for researchers on what the requirements will be when the research is submitted to a journal. And I think there's also things that can be learnt from this in terms of editorial policy wording, instructions for authors, wording to make sure that these things are clear. And I think that the data issues arising after publication also suggests that peer review, if, say, for example, you have cases of fabricated or falsified data, then this isn't something that peer reviewers are necessarily always able to pick up. I think there's lots more interesting information that we could find out in this area, so looking at other fields, looking at the outcome of the manuscripts and the articles that we've provided advice on. Thank you very much.
We do have some questions. Go ahead.
Rob Sebus from New Zealand. As an editor of a small journal, I find your data very interesting. And over the many years, I've encountered the various amounts. I didn't realize really that some of them were bigger problems than others. What I'm more interested in is, what is the percentage of total publications that you receive and the amount of queries your editors send to you?
So I don't have the figures sort of to hand on that, but it is a very small percentage of the total articles that are published that results in any issues, and then I think we're seeing an even smaller percentage of these. I think these numbers are very small in terms of the overall numbers of journals that we're providing support on, and the numbers of articles they're publishing. But yeah, it would be very interesting to look at these as percentages of total publications.
Okay, thank you.
Thank you.
Malcolm MacLeod from the University of Edinburgh. Thank you. Now, for research involving human subjects, there are fairly well-established international ethical guidelines through ICH GCP. And for research involving animals, that's not the case. There's a number of different ethical standards in different domains. And in fact, Retraction Watch carried an example last year of research that had been approved in one domain that wouldn't have been approved in many others. Was that an issue that was live in any of your ethical concerns, or did that not hit your radar?
Yep. So the reason that we've sort of grouped the whole category together of ethical issues is that the number of ethical issues arising on animal ethics issues arising was very small. I would say both in terms of looking at this data, but also anecdotally, by far the most concerns, sort of issues that we receive from editors relate to research involving humans. And I think one of the reasons for that is, as you say, there's much clearer frameworks and much clearer regulations around research involving humans. So I think it's sometimes clearer cut when the editor is concerned about them. We do see sort of issues relating to animal ethics. I think anecdotally that's probably all prior to publication. And as you say, it's normally say an editor reading this and saying, maybe this has ethics approval for the study in the country it was done in, but I don't think it fits international guidelines. I'm not comfortable with that. And then the kind of advice that we would give the editor on that is that if they're not comfortable with what was done and they're not happy with the ethics of it, then they can reject it on ethical grounds, and that we would perhaps suggest following that up with the authors, but maybe also their institution, to kind of say, we understand this had ethics approval, but we have concerns about what was done in the study.
We're running short of time. I do have a couple of questions on Twitter and Facebook. So this is from Twitter. Could increased data sharing reduce issues with data data issues that arise post-publication?
Yes, so I think more eyes. I'm assuming this is saying sort of data sharing before publication.
Yeah.
Because I think one of the reasons that these issues are coming up is that maybe there's not been as much scrutiny as sort of with the numbers of people that are then looking at it after publication. So yeah, I think that could go some way to help that.
And then this question from Facebook. I have observed the same paper published in 2 journals and have read papers with the methods section lifted verbatim from another paper. Are researchers expected to report such issues to journals?
Yes, I think if you spot any concerns on a published article, then yes, do report it to journals, because editors and publishers have responsibilities to the published literature, and if there are concerns about published articles, then yeah, we would take them as seriously as concerns that are raised prior to publication and look into them.
We've just got time for one very quick question. If you have...
Yes, yeah. Irene Hames. It's lovely to see the COPE taxonomy being used, because I should say I was involved in the creation of that 4 years ago. So data, that was the only category that increased on publication. Did you do the analysis, and sorry if I missed this, of which of the areas had increased using the keywords of the different components that constitute data?
No, so we categorized sort of everything that came into our sort of category of data, we categorized as data. So I don't have the sub-analysis on the different types of data issues. I think that's something that would be really interesting to look into. And I know that the COPE taxonomy does go into, you've got a lot of subheadings within the different categories. And I think that's something that would be really interesting to look into in more in more detail than this, because we've gone for quite broad categories.
Yeah, I would guess it would be images. I mean, I don't know if people know about Elizabeth Bick's work, who has been looking by eye, checking 52,000 papers and finding an enormous amount of duplication in the published literature in very reputable journals. So I wouldn't be surprised if data is...
I think. Thank you. Sorry, I'm going to have to. We are encroaching into coffee time now, so I'm going to stop there and say thank you to all of our speakers. And be back here at 3:30, please.